Welcome back! In our last video, we talked about a big question. What exactly is science? We learned that science helps us understand the world through observation, reasoning, and careful thinking. Today, we're going one step further. We're going to explore something called hypothesis-driven science. What is a hypothesis? How do scientists test their ideas? And why is testing so important in science? Let's find out how scientists go from a simple question to a tested answer. They do this using the power of careful experiments and open minds. Scientists establish which general principles are true from among the many that might be true through the process of systematically testing alternative proposals. If these proposals prove inconsistent with experimental observations, they are rejected as untrue. After making careful observations, scientists construct a hypothesis, which is a suggested explanation that accounts for those observations. A hypothesis is a proposition that might be true. Those hypotheses that have not yet been disproved are retained. They are useful because they fit the known facts, but they are always subject to future rejection if, in light of new information, they are found to be incorrect. This process can also be iterative. That is, a hypothesis can be changed and refined with new data. For instance, George Beadle and Edward Tatum first used genetic evidence to propose the one gene, one enzyme hypothesis. This hypothesis was the idea that each gene corresponds to a specific enzyme. As investigators learned more about the molecular nature of genetic information, the hypothesis was modified to one gene, one polypeptide, because enzymes are made up of more than one polypeptide. With still more information about the nature of genetic information, we have come to see that a single gene can specify more than one polypeptide, and the hypothesis was refined again. We call the test of a hypothesis an experiment. Suppose that a room appears dark to you. To understand why, you might propose several hypotheses. The first might be, there is no light in the room because the light switch is off. Another might be, there is no light in the room because the bulb is burned out. Yet another, I am blind. To evaluate these hypotheses, you perform an experiment designed to eliminate one or more of the hypotheses. For example, you might test the first hypothesis by flipping the light switch. If you do so and the light comes on, your test disproved the first hypothesis. Something else turned off the light initially. If the light doesn't come on, you test other ideas. Note that a test such as this can rule out a hypothesis, but it does not prove the others are true. It only demonstrates that one is false and the others remain possible. A successfully experimental hypothesis yields predictions that are consistent with the results and is thus retained. Often, scientists are interested in learning about processes that are influenced by many. To do so, they use controls, experiments where all variables are held constant except one. This is done to determine which variable is responsible for any observed changes. Controls eliminate variables other than the one being tested. In all other respects, controls are treated exactly the same as the experimental group. This helps to ensure the experiment is fair and accurate. A successful scientific hypothesis needs to not only account for what is already known, but also be testable in a way that shows incorrect predictions are false. The predictions provide a way to test the validity of a hypothesis. These predictions are often made using general principles as a guide. The general principles are called deductive reasoning. The predictions made using hypotheses are then tested through experiments. For example, based on early studies of microorganisms, scientists once predicted that messenger RNA, mRNA, transports the genetic information from DNA to the protein-making machinery of the cells. Scientists used the idea of labeling mRNA and ribosomes to test this prediction. Two hypotheses were proposed. One hypothesis said that both labeled molecules go to the same places in the cell. Another said if mRNA is not the message carrier, they would show different movements. The results supported the first hypothesis, supporting the gene expression theory. Thanks so much for joining me. Isn't it amazing how much wonder is hidden in the things we often take for granted? Take care, stay curious, and I'll see you in the next chapter.